So, I show some examples. Okay, this is one sequence for a particular protein, and he gives something on the red font. So, can you see this red font? So, can you guess the region with the red font?
Hydrophobic.
It is completely hydrophobic, right? If you see the residues, most of the residues are alanine, valine, phenylalanine, isoleucine, so on. In this case, this protein, this region is predominantly with the hydrophobic residues. So, I give another sequence, right? This is another sequence called 5-CRO. So, if you see this sequence, this is short sequence. By short sequence, you will get the predominance of some specific residues. Which residues are dominant?
Positively charged.
Positive charge residues. So, how many residues here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 residues, right? 11 positive charge residues in this sequence. Here I have another sequence. Here if you see, there is a higher occurrence of serines. You can see several serine residues in this case. So, when you scan the literature, if you see the literature about the functions of different proteins, right. So, we can guess, okay, these positive charge residues are important to interact with negative charge moieties. In this case, you can think of a protein which can have high tendency to interact with negative charge moieties. So, which molecules have negative charge? Mainly DNA is negative charge because of phosphate group. So, in this case, it requires the opposite side, positive charge residues. So, this protein could be a DNA binding protein, right, have high tendency to interact with the DNA. In fact, yes, because 5-CRO is the CRO repressor, right, this interacts with the DNA. Likewise, here if you see, so here you can see the dominance of a stretch of hydrophobic residues. These residues prefer to be in the membrane, right. So, in this case, this chain could be a membrane protein, right. Actually, that is correct because this is a photosynthetic reaction center of M chain. So, in this case, this is a membrane protein, right, it has several transmembrane segments, this is one of the segments. So, likewise, here you can see one example. Here this is the sequence for one beta barrel protein. Beta barrel proteins have high dominance of serine residues to form the hydrogen bonding network to maintain the stability and the function structure of these barrels, right. So, this is why it is highly occurring in this particular protein, fine. Now, the question is, if I give these 3 sequences, is it possible to discriminate? Okay, A is this type, B is this type and C is this type, yes or no? Okay, let us see. So, there are various features we discussed. What are various features we discussed till now? Composition, occurrence, molecular weight, hydrophobicity and different amino acid properties, right. So, for example, if you have 2 groups of proteins, group A and group B, if I give you a new protein, can we able to tell this protein belongs to group A or this protein belongs to group B? Okay, so, if here I take 2 groups, group A and group B, so I get the for example, I collected all the proteins in the uniprot in group A and the proteins belong to group B. Then if you get all these sequences, I can calculate the composition, right, because just you can calculate n of I by n, take all the residues, count the residues of each type, normalized by n, you will get the composition, right. This is group A, for example, this is group B, you get the composition for 20 different residues, here 10 residues and here 10 residues. And when you compare these 2 values, for example, if you take alanine, for the group A the value is 8.47 and the group B 8.95. If you get the deviation, right, then you can, this is almost similar, right, this is average value, but if you consider the standard deviation, so this will be plus or minus 0.1 or 2, right. So, in this case you will get the similar numbers. Some cases yes, for example, aspartic acid there is no change at all, here 5.97, right, here is 5.91, there is no change, the difference is only 0.06. But some residues, if you look in the details, they are highly different. For example, if you take cysteine, here it is 1.39, but here in this case it is 0.47, there is 3 times different, 3-fold difference, 0.5 and 1.4. Likewise, if you glutamic acid, histidine, right, this is 1.25 is 2.3, 2 times difference, right, isoleucine. I will show some of the more data, these other residues. Here also you can see the difference, it is other way around, here it is highly dominant in the case of group B, this is 8.05, this is group A, it is only 5.9, this is 6. So, some residues which are similar in both the cases and some residues which are significantly different, right. If they are different, then we can use these residues as the features or the parameters or the properties, right, to discriminate or distinguish between these two groups, group A and group B. So, how to do this? First we see the important residues, and some residues is just close to the 0, this is a difference of composition. I calculate the A minus B, right, composition of A minus composition of B, right. So, we have the average values. So, if you some cases, okay, it is very high, they are less than 0, and some cases they are above 0. So, if you look into these different residues, I made a few groups. This is the aliphatic and the second one aromatic here. These are sulfur containing residues and polar residues and charged residues, and we could see some patterns. Some cases we can see pattern, for example, the polar residues. All the polar residues which are highly preferred in the case of this group B, because minus. Some of these the sulfur containing residues, they are dominant in the case of group A. Here you can see some cases, right, also mainly charged residues in group A. So, in this case you can try to relate, you can try to understand why some residues are dominant in group A and some residues are dominant in group B. Actually, we know what is group A and what is group B, the structures we know, the functions we know. So, now we can explain the importance of these residues, why its occurrence is high in some cases, why it is low in some cases, right, we can find. And we use this information if it is very highly different, for example, this is highly different, even this is plays important role for discrimination. So, if you take these residues and the combination of these specific residues, we can able to distinguish between the proteins group A as well as group B. Fine, how to do this? So, it is a very simple algorithm, right, but you can complicate the algorithm, right, when you refine the results. Simply what we do, we have the set of proteins from group A and group B, take all the proteins from group A and the value of group A, composition you can calculate, then take all the proteins from group B, get the composition, right, we have two set of values. Okay, for example, here we have the data, one is A, one is B. Now what you do is we get the new protein X, we do not know this belongs to A or this belongs to B, right, A or B, we do not know. Then what to do? First get the composition of this particular protein. So, we got 20 numbers here. So, here we have 20 numbers, right, we have here 1 to 20 for 20 residues and composition B 1 to 20 and protein X also we have 1 to 20. Now, we take compare the each composition, for example, A, D, C or 20 residues, get the difference, composition of A minus composition of this X, respectively we have to get the difference, take the absolute and repeat for the 20 times 20 residues, then get the summation I equal to 1 to 20, and finally we get a number, this we can say this sigma A, deviation from the group of proteins A. Repeat the same, take the same composition X and compare with B, do it for 20 residues I equal to 1 to 20, right. So, get the difference, now you can see the total difference will be sigma of B. So, with the comparing composition A we get sigma A and comparing with the composition B we will get sigma B. Now, compare these two, sigma A and sigma B, which one has less deviation, which one is less deviation here, B is less, then this belongs to group B. If A is less, then the protein X belongs to group A. So, here just we compare the deviations. We can also apply some error functions, right, we can apply error function sigma like 0.1 or 0.2 and 0.3 so on, and you can optimize the performance. You can, I will explain later about the how to assess the performance. There are various measures to assess the performance like sensitivity, specificity and accuracy, I will discuss in later classes. So, you can add these sigma and see in a set of 100 proteins we are able to predict or discriminate exactly for the maximum number of proteins. then you can define this error function. In this method we use the composition and the deviation. Instead of deviation you can also try to use some other measures. What other measures you can use? Here you use deviation, we can also use the correlation. We have 20 numbers here, 20 numbers here, right. You can get the correlation with A. Likewise, you can get the correlation with B, right. Then how, which one you need to select, which is if it belongs to A, the correlation will be higher, right. If this correlation R with the correlation with A is 0.9 and R with B is 0.7, then the protein belongs to A, right. So, you can use correlation, you can use the deviation, and you can use any error functions and so on. Also here now we use the composition of all the 20 residues, but if you look into this figure, all 20 residues are not showing very high difference, only some residues the difference is significant. Then you can try with the residues which are showing only significance between this group A and B and see whether we can able to discriminate better or not. Secondly, when you develop any method, it is important to reduce the number of parameters, number of properties. Instead of 20, if you could get the same performance with 10, then 10 is better, right. Unnecessarily we do not have to include noise in the prediction performance. Okay, now I will show an example. Okay, here this is, I consider two proteins, one is the from group B, this is group B. Okay, here this is from group A. So, I have the sequence. Now, what to do? Get the currents. Okay, here this is the N is here. From this N you can calculate the composition, this composition. Then from this composition we know the composition from the globular protein or group A or group B, right. You can see here. Okay, this is the composition of group A and composition of group B. So, then get the subtract the values. So, this is sigma of group A and sigma of group B. Do it for all the residues, then finally sum up. Here this is 34 and here 39. Which one is small? This is small, right, this is small. So, this belongs to group A. Then take the example group B. Here also we have calculated the N. From N you can get the composition, and here you can see this A, this B, get the deviation, right. The original values we have here, these are original values. You take the composition and subtract from either A or B for the 20 residues, right. So, 8.47 is here and you can see this is 8.31. So, the difference is 0.16, for the other case it is 0.64. Likewise, you can see the difference mainly if you see the serine is here, is 8.46. 8.46, if you look into this A, it is 5.94 and the B it is 8.05, right. You can see the difference here is 2.52 and B it is 0.41. Now, take the summation. So, this is 23, this is B, which is small, B is small, right. So, B is small, so this is identified as group B, right. So, this is simple algorithm, but we need to refine this, adding various other factors and estimating the performance and validating, right, that everything we need to do, but simple algorithms or simple features we obtain from the sequence, you are able to classify the proteins from different structures or different functions. So, then once is that we can do several online methods. So, here these are the various methods you can discriminate these types of proteins group A and group B. So, if you go with the composition, you can try with the composition, right. So, if you give the sequence, right, here you give the sequence here, right. If you click on this, then you can give the sequence. If you get the sequence, then it will automatically calculate the occurrence. From the occurrence you will get the composition. From the composition you will get the sigma of A, here sigma of B, and from this number we can see this is group B. So, it belongs to group B, I put this beta barrel membrane proteins as group B, right. So, if you see this one, the difference is very less, because if you add several residues and if you add only use the data only for specific residues, you can improve the performance, that we need to try again and again. So, improve the performance. Okay, I have a question. So, this is the data, So, I have the composition for alpha, I have the composition for beta, right. I have two values, this alpha composition and this is the beta composition. So, now I have a question. So, this is the question, I give this sequence, I want to know whether this belongs to alpha or beta. How to proceed, what to do?
Composition.
Right, first we use the sequence and calculate the this is the composition, right. I can calculate, okay, take number of A's, number of D's and normalize with the number of residues. Okay, this is for the same order we give, right, G A V, right, this is G, this A, this B and so on. Now what to do? Take this number and subtract with these numbers alpha, right. So, if we add up for all the residues we get 20 numbers, then we take the absolute values and add up, if you add up everything then you will get this number. Then what you have to do? Repeat the same with this number, this beta number, and for 20 residues we calculate and sum up, right, the absolute values, finally we get this number. Then what you have to do? You have to compare these two. So, and then how to decide is alpha or beta? Which are lower, right? Because the deviation lower, that is biased to that particular protein, right. So, this is lower, so we can predict this as alpha. I give another question here, you can do the exercise. So, this also you can you have a sequence, this is a long sequence, with small sequence you can easily count, long sequence you have to write a code, right. You can calculate as we discussed earlier about the algorithm, we can write a program to calculate the composition. So, this is the result, 20 residues we have the composition. Now, you subtract this from either alpha or from beta, then you have to get the absolute values and take the summation, right. You will get the values, alpha is 36.09, beta is 18.63. So, if you compare these two, right, this is less, so this belongs to beta. So, if the difference is significant, very high, 10, 15 difference, then easily you can discriminate. If it is very close, then it is difficult. Some cases, this is why all the prediction methods sometimes fail in some aspects. You cannot get 100% prediction accuracy, because you can see some type of overlapping, like as you see in this composition, right, you can see some cases it is very close. So, it is very close, maybe either group A or group B, this is the reason why I find it difficult to discriminate. For that cases, we have to identify some features, right, not exactly this one, which can discriminate in some other way, only at least to that situation. In this case you can combine, only you use with very large difference in composition you can use this, if it is less difference you use some other features, right, then you can incorporate. In this case you can improve the performance, one gives 70% and another aspect will another 70%, right. If you combine both you can increase to 80%, right, you can do that. So, now there is another features, right, there is pair preference. In the composition we used only one residue, and see how many times A, how many times D, how many D, E, all these things. But then the question is the occurrence of A always close to A, always next to D, always next to E, right. Here there are two A's, this A is just before C, this A is maybe something else, for example this L. So, this A is after C, this is after A, but this is after L. So, now the question is how many times A comes next to A, how many times A comes next to C or E and so on, right. In this case you can get the pair preference, because we did two peptides. So, this is why we call this as dipeptide, right. If you take these two, so we can say this is dipeptide, like, and these two, this is AC and here this is CA. So, in this case there are various ways to get the preference. So, if you see the dipeptide of I comma j, then you can see Nij with number of times of residue I which comes next to j, I and j are the distribution of the 20 residues, and to get the percentage you get 100, right, but you can normalize with the different factors, either you can normalize with n as we did in the case of composition. Here I normalize with Ni plus Nj, that means totally how many residues which are involved in this I and j. If it is I 5 times and j 9 times, then there are 9 cases in the sequence. So, I normalize Ni plus Nj, but if you want to get the probability, right, if there are I residues of type I and another residues of type j, n residue type j, then you can use as Ni into Nj. So, there are different ways to normalize, but they are related to each other, right, but eventually the one which gives the importance is Nij, number of times, right, the residue I which is come close to j, right. This is distribution of residues, right, at the positions I and I plus 1, right. Here Ni is the number of residues of type I and Nj is the number of residues of type j. For example, if you use this equation and I give a peptide, so if for example if you see dipeptide preference for Pe, how many times Pe occurs here? 1, 2, that is it, 2 times. How many P's and how many E's? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 5 P's and 5 E's, so is equal to 10. So, you can get this value will be 20. Likewise, if you see this real protein, okay, so here you can see number of AAs, right, totally how many times A comes 1, 2, 3, 4, right, 4 times AA, but total AAs is 17 times. So, if you get this, you will get the value of 11.76. If you see, interestingly, if you see other case it totally 0. If some two classes, one class it is 0, another class if you get some numbers, then we can say that pairs are important, that pairs can be able to discriminate in different classes. In this case it can perform better than the specific composition, right, just we use only one residue preference for any particular protein. So, in this case dipeptide proteins preference more information, but the drawback is there we have 20 values, but here we get 400 values. So, if your data set is very high, then it is good to use the residue pairs. Data set is less because we get several zeros, but that is not so good for that discrimination. So, in this case you better reduce the number of pairs or reduce the amino acid to amino acid composition. So, summarize, what did we discuss today?
Lot of features.
Different features, right. If we have a sequence, what can we do? What are the different features we discussed? Composition, occurrence, molecular weight, hydrophobicity, different properties, pair preference and so on. Then also we explained one example to distinguish between the two types of proteins, group A and group B, using amino acid composition. There are several ways we can use the several features, right. You can use for the distinguishing different types of proteins based on structure or function and so on. Next class we will discuss about the more details about the hydrophobicities and how to construct profiles, right, and you can use the potential applications of the different aspects from the features obtained from the primary sequence. Then we go with the secondary structures and secondary structure prediction and so on. Thanks for your kind attention.